Today we're going to talk about protein synthesis. Now, protein synthesis brings in this central dogma of DNA. Now, the central dogma of DNA is as follows. DNA, as we know, is the storage for genetic information. It stores genetic information. And DNA is our blueprints, or it codes for all of the instructions and all of the genes that make up all the different traits that you express and makes up who you are. So DNA has our instructions. The central dogma of DNA is DNA then goes to RNA, which, as we know, transmits genetic information. And then RNA, we're going to find out, ends up producing or helps to produce proteins. So this idea that we go from DNA to RNA to proteins is referred to as the central dogma of DNA. Now, as we discussed before, when we talked about macromolecules, proteins do a whole lot of things in our body. They make enzymes. They make structural components. They're found in the cell membrane. So the proteins do a whole lot. So we really wouldn't be here without our proteins. So we're going to talk about how we end up making proteins. And we make up proteins through a two-step process. The first step is transcription, which takes our DNA to RNA. And the second part is translation, which takes our RNA to protein. So let's get started. So again, protein synthesis is a two-step process. So our first step is transcription, our second step is translation. So take a look at this diagram. Here we have a diagram of a eukaryotic cell. Within the eukaryotic cell, we see here is our nucleus, and within the nucleus we have our DNA molecule. Now, transcription takes place in the nucleus. That is important. That's something that you should write down in your notes. So transcription takes place in the nucleus of a cell. And transcription literally means to rewrite. When you're transcribing something, you're rewriting something. So we are rewriting DNA into the language of RNA. So that's transcription. We are rewriting DNA into the language of RNA. DNA and RNA are both nucleic acids, so we're not making a big change, but we are making a change. That RNA molecule is then going to move out of the nuclear pore and move on into our cytoplasm. That RNA, once we're in the cytoplasm, will meet up with a ribosome, like so. This is my lovely little ribosome here. And our second part of protein synthesis will take place, and that is translation. So translation takes place at the ribosome. So in the cytoplasm at the ribosome. So there we go. We've got the ribosome there. We also have other RNA molecules involved in translation, which help us make our final end product, which is the protein. Now, translation is when we are taking this RNA and translating it into a protein. So when you translate something, if you think about the definition for translate, you usually take something like the language English and change it into something completely different like French or Spanish. So that's exactly what we're doing with RNA or mRNA to our protein. Is we're taking RNA and we're translating it into something completely different. We're taking an amino acid sequence and we're building ourselves a protein. We're no longer a nucleic acid. We're now in the language of proteins or amino acids. So this is just a brief overview of protein synthesis. Again, it's built up of two parts, transcription, which takes place in the nucleus, and translation, which takes place in the ribosome. In transcription, we're going from DNA to RNA, and in translation, we're going from RNA to protein. So there are three types of RNA that you need to be familiar with. The first type is mRNA, and mRNA stands for messenger RNA. And messenger RNA is responsible for carrying the instructions to make a protein. So since DNA is such a huge molecule, and it's such an important molecule, it has all of our instructions, we don't want DNA to leave the nucleus. So what we're going to do is we're going to make a copy of the gene that we want, and we are going to transcribe that into mRNA. So that mRNA is like, think of mRNA as like a photocopy of the gene that you want. And that mRNA is going to leave the nucleus, and it is going to bind with a ribosome. Now, our ribosome is made up of our next type of RNA, and that is rRNA. So rRNA is part of the structure of the ribosome. And remember that ribosomes are involved in making proteins. They're the site of protein synthesis. See, this all links together. If you memorized your organelles, you know all about the ribosome and the function of the ribosome. So our RNA is part of the ribosome. Once that messenger RNA docks with the ribosome, then we have our last type of RNA, our transfer RNA, which is responsible for transferring amino acids to make a protein. So here's our tRNA molecule. It sort of looks like a T, if you will. So, if you just drew like a nice little T, it kind of looks like a T. And a tRNA is responsible for bringing in amino acids. So an amino acid, depending on the type of amino acid, will be attached here. So here would be an amino acid. I'm abbreviating AA for amino acid. So amino acid would be found right here, and you'd have a variety of different tRNA molecules with each of their own different amino acid coming and bringing and reading this mRNA molecule and bringing in the correct amino acids to build our protein. So let's take a look at the steps of transcription. So first off, we have RNA polymerase. Okay, RNA polymerase is an enzyme, again, because it ends in ASE. And RNA polymerase binds to a gene. So a gene codes for a specific trait. So remember that a gene, codes for a trait, whether that be hair color, eye color, etc. So a gene codes for a specific trait. It could be the gene for lactase, like your body wants to build the enzyme that breaks down lactose because you're drinking milk and you need to break that down. So again, a gene codes for a trait. So RNA polymerase, that enzyme, is going to bind to the two strands of DNA, unwind it, and separate it. So it does the job of helicase. If you remember back to DNA replication, RNA polymerase does the job of helicase as well. RNA polymerase also then adds complementary RNA nucleotides to one strand of the DNA molecule. So we're only copying, if you will, one strand of the DNA molecule. So complementary RNA nucleotides are added and joined to, we're going to say, one strand of DNA, and that strand is referred to as the template strand. Okay, when RNA polymerase reaches the end of the gene, it hits a termination signal. It's a number of bases, about 3 bases that signal for the RNA polymerase to drop off and release both the DNA and the mRNA. So let's take a closer look at a diagram of what this looks like. So here is a diagram of transcription. So remember, in transcription, we are doing that first part of the central dogma of DNA. So we're going from, in transcription, we're going from DNA, that first part of the central dogma, to RNA. Okay, so here we have RNA polymerase right here binding to a gene's promoter region, and the promoter region is just the start region of the gene. That RNA polymerase is going to unwind and separate the two strands of DNA, and then it is going to proceed to add complementary nucleotides to only one strand of the DNA. And remember, this strand, the strand where it's adding those complementary nucleotides, is referred to as, again, the template strand. There we go. So we've got our template strand. Next, we got to take a look at the genetic code. So once we have constructed our mRNA molecule is made up of a sequence of bases, A's, U's, C's, and G's, okay? And we can break up those bases into groups of 3. And a group of 3 bases, so a codon, is a group of 3 bases, like A, U, G, for example. Okay, so a codon is a group of 3 bases, and a codon represents or codes for an amino acid. That's why it's called a codon. A lot of amino acids have more than one codon. There's 20 amino acids, but there are 64 codons. So clearly, a lot of amino acids have more than one codon. There are some codons that do not code for an amino acid at all, and they code for a stop. Okay, that means to stop translating, and the protein is complete. So to read the codons on an mRNA molecule, we use a tool called a codon chart. Here's an example of a codon chart. You have one with you. If you don't have one or you misplaced yours, you can find it up on the locker on School Loop. But you read a codon chart, or this particular codon chart, from the center outside, so from the inside out. So for an example, if I'm reading an mRNA molecule, I break it up into a group of 3. That's our codon. This codon AUG, if I'm going to read that, I go A from the inside, there's my A. Then I go U, here's my U, and then I go for G, and there's my G, and then we see that the codon AUG codes for the amino acid methionine. When you do write the amino acids, you don't need to write the full name of the amino acids. They're quite long. All you really need to do is write out the first 3 letters of the amino acid. So codons and anticodons. So codons are found on mRNA. Anticodons match up, are found on tRNA, and they are complementary to the codon sequence on the mRNA. So how does a tRNA molecule that brings in the amino acids to build a protein, how does tRNA know that it's bringing in the correct amino acid? Well, it knows it's bringing in the correct amino acid because the anticodon matches up complementary to the codon on the mRNA molecule. So translation. Let's take a look at the steps of translation. So now that we know about our mRNA and our tRNA, let's look at the steps of translation. So once an mRNA molecule leaves the nucleus, it's going to go into the cytoplasm, and it's going to bind with a ribosome. That mRNA is going to bind with a ribosome, and then a tRNA bearing the amino acid methionine is going to bind together as one. That's that first step. Then other tRNA molecules carrying amino acids specified by the codons on the mRNA are going to start to arrive. Peptide bonds are going to form between adjacent amino acids. So once the tRNA has brought in the correct amino acid, a peptide bond, that's a specific bond between two amino acids, a peptide bond is going to form between two amino acids that are next to one another. Once that peptide bond has formed, the tRNA molecule is going to leave and go find itself another amino acid, and a new tRNA molecule is going to move in, carrying the amino acid for the next codon. And this process is going to repeat itself over and over again until we reach a stop codon. There's only 3 stop codons, so until we reach a stop codon. Once a stop codon is reached, the ribosome falls apart and the protein is released. So the ribosome will fall off, the mRNA molecule will fall off, and the tRNAs will fall off. So let's take a look at some diagrams that illustrate this. So here we have translation, okay? It starts out with the initiation process. So when the ribosome subunits, the mRNA and the tRNA carrying methionine bind together. So here, right here in this diagram, we're seeing that initiation sequence taking place. We've got our ribosome that's binding to our mRNA. We've got our tRNA with our methionine binding to it, and we've got our mRNA being read by our tRNA. So that's our initiation. That's the first step in translation. The next thing is elongation. So the tRNA carrying the amino acid specified by the next codon binds to that codon. So we can see that here. And a peptide bond forms between adjacent amino acids, and the ribosome moves the tRNA and the mRNA. So it's sort of like a little conveyor belt, if you will. So we've got this original tRNA, and then the next tRNA comes in, and then a peptide bond is formed, and then the tRNA will leave, and then we'll bring in a new one. So it's a little conveyor belt that starts to happen. Elongation is going to continue, so we've got tRNA molecules coming in. We've got tRNA molecules that have dropped off their amino acids leaving, and this process takes place over and over again. So the first tRNA detaches and leaves its amino acid behind. Elongation continues, and the polypeptide chain continues to grow. So here we see the polypeptide chain continuing to grow as tRNA molecules are bringing in their appropriate amino acids, and the tRNA molecules are leaving. Once a stop codon has been reached, that's the termination process. So once the stop codon has been reached, there is no tRNA that codes for a stop. So pretty much when a stop codon is reached, there's no tRNA that's going to come and arrive there. So that signals to the ribosome, the mRNA, and the tRNA that it's done making the polypeptide. So then we get to disassembly, where the ribosome complex falls off and the newly made polypeptide is released and ready to either be processed further in the endoplasmic reticulum or gets shuttled to the Golgi apparatus, where it gets packaged for export outside of the cell. So just to recap, you should be familiar with the 3 different types of RNA. And remember, that's the 3 different types of RNA are mRNA, which stands for messenger RNA, rRNA, which is ribosomal RNA, and ribosomal RNA just means that it's found as part of the ribosome. And then we have our tRNA, or our transfer RNA, which is responsible for transferring those amino acids over to the ribosome and reading that mRNA molecule, the codons on the mRNA. You should also be familiar with the steps of transcription and translation. Know that transcription is when we rewrite DNA into RNA. Okay, and translation is when we take RNA and translate it into the language of protein or amino acids. You should know where transcription takes place. Know that it takes place in the nucleus. And you should also be familiar with where translation takes place. Know that translation takes place at the ribosome, in the cytoplasm. Okay, so make sure you know this information. If you have any questions, be sure to fill that information out on your form. Have a great evening. Bye.